All right. Good morning. Here we are. Web-based, web-based learning. All right. So good morning. This is kind of new to me, and probably also to you. But we're going to try to deliver the lessons this way for the time being. So what I want to do is I want to look at Lab 9, which is the urinary system, and the first thing I think that we should do is look at the lab atlas and go over that stuff, and then we'll come back here to the actual lab paper and we can fill out some things together, and then I'll probably leave you with some independent things to work on. All right, so let's see. Let's go down here to the lab atlas. Here we are. And this starts off with the major organs of the urinary system being the kidney. And of course, the job of the kidneys is to filter the blood, of cell wastes. As we go throughout our day, our cells are working and creating wastes, right? And these wastes end up in the blood. So it's the job of the kidneys to receive this unfiltered blood and filter it from excess waste products, and then reintroduce the blood back to the body filtered. And the kidneys are the major organs for that. So if we look at the structure of the kidneys, we could see that all of this on the outside, which has a lighter color to it, is the cortex. Inside of that, deep to that, we have what's called the medulla, and you can see the medulla contains these pyramid type structures. These are called renal pyramids. And then, of course, we have a major blood vessel entering into the kidney. This is called the renal artery, and that brings unfiltered blood in. And then we have the renal vein. After the blood is filtered, it's deep inside the kidneys. The blood exits the kidney through the renal vein. And of course, as blood is being filtered, it's producing urine. So as the urine is being produced deep in the kidneys, the urine drains down through these specialized structures into this area over here. And we call this the renal pelvis. And the renal pelvis leads to the ureters, which is that structure there. All right, so that was a model. This is a real image of a real kidney, probably a sheep kidney. And again, the cortex is everything on the outside. Deep to the cortex, we have the medulla that contains the pyramids, and the collecting ducts all converge to this middle area called the renal pelvis, and the renal pelvis drains all of the urine out into the ureter, which is that there. All right. Moving on. Now, inside the kidneys, we have millions of filtering units. These are called nephrons. The nephrons are tubular. And what happens, there's an interaction between the blood vessels and the nephrons. So the blood vessels enter the nephrons, and the nephrons, like little tiny vacuums, absorb all the waste products, including water, nitrogen compounds like urea, excess salt, but also good things like amino acids and glucose and stuff like that. So everything is getting taken out of the blood in the very beginning, but then most of the good things get put back into the blood, and all those waste products end up collecting in the collecting tubules and then get eliminated from the body. If we look here, again, everything on the outside usually has a lighter color to it, is the renal cortex. Everything deep to that contains the pyramids. We refer to that as the renal medulla. You can see some of the nephron parts extend down into the medulla, and also the collecting tubules extend down into the medulla. Here we have a very important part. This is as the renal artery enters into the kidney. What happens is the blood will get directed towards this structure here. This is called a glomerulus. And the glomerulus is a knotty arrangement of capillaries. And what happens is as the blood is going around and around in this knotty arrangement of capillaries called glomerulus, it forces the contents of the blood out if those contents are small enough. So some of the larger things like red blood cells will not leave the blood, nor would we want them to. And other large molecules like proteins and stuff like that are too big to exit right here. But most of the small things will. So any excess water that's in there, salt, amino acids, um, urea, a lot of these different types of things are going to be exiting the blood here at the glomerulus. And when they do exit the blood, they enter into this cup like structure that surrounds the glomerulus called the glomerular capsule. And then the glomerular capsule takes all that stuff that are going to call filtrate. Now, filtrate is going to be eventually going to be urine, but it's not quite urine yet. So anyway, the glomerulus empties the contents of the blood into the glomerular capsule. From the glomerular capsule, the filtrate finds its way to the proximal convoluted tubules. And a very important process happens here. This is the process of reabsorption. So remember, just a minute ago, everything's coming out of the blood and emptying into the glomerular capsule, but it's in the proximal convoluted tubules that reabsorption is going to occur. So some of the good things that were taken out of the blood are going to be put back into the blood. And then from here, the filtrate goes down into the descending loop of what's called the loop of Henle, and you can see we go all the way down into the medulla, and then up the ascending loop. And when we're going down here, this area over here is hypertonic compared to this area over here. So this is the section of the kidney that is used for maintaining salt and water balance. So then the filtrate will extend back up towards the cortex and eventually it will find its way into the distal convoluted tubules. There's another important process happening in the distal convoluted tubules. It's called secretion. So this is where the blood will drop off anything that it couldn't drop off earlier. So for example, larger items. If you're taking prescription medications or you're experimenting with recreational drugs, it would get sent into the urine directly from the blood through the distal convoluted tubules through the process of active transport. And then those items would be added to the urine, and then finally the urine would all collect in this structure here called the collecting duct, which would, and you could see it's not one nephron to one collecting duct. There's one collecting duct that receives urine from many, many nephrons. And then all the collecting ducts would converge into the renal pelvis. So here, this is just a different angle. We're looking at just that one nephron now. You can see the renal artery brings unfiltered blood in, and that blood would spin around and around through this structure called the glomerulus. So, but it's the afferent arteriole that takes the blood directly from the renal artery and sends it into the glomerulus. After the blood has been moving around, spinning through the glomerulus, it will then leave this capsule, blood, having lost a lot of its contents, and it will go through what's called the efferent arteriole, and then you could see it will make its way through the peritubular capillaries. So as the filtrate is going through the proximal convoluted tubules, down the descending loop of the loop of Henle, the ascending loop, and then the distal convoluted tubules, the blood is kind of going all over the place, picking up a lot of different things that it had lost back here during filtration before the blood exits the kidneys completely filtered through the renal vein. All right. So this is a microscopic image. This is what you would see if we were looking at this using our microscopes in class. This is a low power view. So you can see the objective lens would be 10x the total magnification. When we include the eyepiece, would be 10x, giving us a field diameter of 1,800 micrometers. So what are we looking at? Well, we're looking at the knotty arrangements of capillaries called the glomerulus, surrounded by this clear space. This clear space is called the glomerular capsule. So there's one. Here's another glomerular capsule with a glomerulus inside of it. And another one. Again, every kidney would have millions of these, enough of these to do the job of filtration. Just with one kidney, you only really need one kidney. This is a high power view, so 40x objective lens. Zooming in, now we have a field diameter of 450 micrometers, but you could see there's that knotty arrangement of capillaries called the glomerulus, and we see this clear space that surrounds it. So as the unfiltered blood is spinning around through here, the items that are leaving the blood are filling this space here, right? This glomerular capsule, and that's going to be, eventually will be urine. So the glomerular capsule surrounds the glomerulus. And then, of course, what lines this space is simple squamous epithelial cells. You can see they're highlighting the nucleus of those cells, right? So let's take a look at the lab. Maybe we can fill out some of the paperwork together. So lab 9 urinary system. Identify the parts of the urinary system and the kidney. Learn the parts of the nephron, and we're going to trace the fluids through the nephron. We're going to compare the contents of the blood, the glomerular filtrate, which eventually will become urine, and then urine. In this lab, there's a test called urinalysis that we would be doing, which I don't think we're going to be able to do using this method. So I'll probably put a video up of somebody performing that. All right, and then it says identify a kidney slice. Well, that's what we just did. We just looked at those images under the microscope. So we'll look at that in the lab, and I'll tell you what I want from there. But we could kind of fill in some of this stuff. What is the functions of the kidney? Well, all right, that's kind of easy. The kidney are the major organs of the urinary system. Sounds good. And the ureter, what do the ureters do? Well, the ureters connect the kidneys to the bladder, so they direct urine from the kidney to the bladder. You could be filling this out in your book as I'm doing it. And of course, the urinary bladder, what does that do? That stores urine. And the urethra directs urine from the urinary bladder to the outside. Now we have a renal artery. This is in red. Renal vein, that's in blue. Remember, when we see red and blue, that doesn't always mean that the red is an artery and the blue is a vein. It just, in the models, it usually means that the renal arteries, or any artery, this blood vessel would be carrying oxygenated blood. And if it's in blue, most veins are written in blue, except for the pulmonary vein. But if it's in blue, then that indicates to us that it's carrying deoxygenated blood. All right, so Let's fill that in. The renal artery brings unfiltered blood into the kidney. The renal vein, we can say, brings filtered blood from the kidney back into circulation. Sounds pretty good. And so basic functions of the different structures. There's some labeling that we could do, but before we do this labeling, let's just write some basic descriptions and functions of the cortex. The cortex is the outer layer of the kidney. Medulla is deep to the cortex, and it contains the pyramids. I'm not going to write that, however, because it does say that there. The renal pelvis is the site where the collecting tubules converge. And then, of course, the ureter, we already did that one. We don't have to do that one again. Renal artery, we did that one too. And renal vein. But what we can do, let's scroll down a little bit, is we could label this. All right, so we have the renal cortex here. And we have the medulla just deep to that. And we have the renal pelvis. Over here we have the renal artery. Didn't work. Did it? Oh boy. What happened there? What did I just hit? What did I just hit there? Why did that go sideways? All right, I'm gonna have to pause this for now and figure out what oh, here I did this, I think. No. Let's try one more. There we go. Let's move back up. All right, let's try that again. There's our renal artery. I want this over here. Oh, well, let me go over there. Come on. Are you kidding? Anyway, how annoying. Well, let me go over there with that. Anyway. No. That's a bummer. I wonder why it's doing that. Maybe if I minimize. Let's try that. was no good. All right, let's try this now. All right. Maybe that's a little bit better. No artery. Good. Go a little bit bigger, maybe. All right. And below that is the renal vein. And you can see this is a new process for me, but most people would stop the video because they made a mistake and start it all over again. We're not doing that. We're just going to get through this as if this is live, right? Let's move that over there. And this last thing is pointing to the ureter, so let's fill that in. All right. I feel like we'd be taking this long if it was me doing this in class anyway. All right, the nephron is the basic unit of structure and function, and that's exactly what we're going to put here. The nephron. All right. The nephron produces blank in the process of removing waste, and here what they're talking about is urine. It produces urine. All right. And there are about 1 million nephrons in each kidney. Okay. So what are we going to do about these descriptions here? What do we have here? We have tubular parts, and then we have vascular parts. So the tubular parts are the parts of the nephron. So as the blood is coming into the kidney, the tubular parts are collecting the contents from the blood and are going to be removing those contents. The vascular parts are going to be the blood vessels that are involved in bringing unfiltered blood into the nephron. And then after the blood is filtered, allowing that filtered blood to exit, bringing that blood back in circulation. So if we go through the tubular parts of the nephron, this is a good opportunity for us to kind of go over the 3 major processes that are happening. One of them is happening between the glomerular capsule and the glomerulus, and this process is called filtration. So this is the site of filtration. What that means is everything is coming out of the blood. If it's capable of leaving the blood, it's coming out. So if it's small enough, it's coming out. But remember, good things are leaving the blood here too. All right, so that brings us to our next major process, which is called filtration. This happens in the proximal convoluted tubule. So this is the site of reabsorption. Reabsorption. Now, what's reabsorption? That's where the blood is taking some of the good things back, taking some of that water back if it needs it, taking some of the glucose back. In fact, all of the glucose is going to be put back. Some vitamins, amino acids are getting put back. All right, so the loop of Henle, this is a site salt and water balance between the blood and the urine. We have the descending limb. The descending limb brings the filtrate down into the medulla, and the ascending limb brings the filtrate back up towards the cortex. The distal convoluted tubules is the site of our 3rd major process. This is called secretion. And this is where the blood drops off. It's last minute items that couldn't drop off before. Things like prescription drugs, steroids, recreational drugs. antibiotics. If you're on antibiotics, they end up in your urine. At the distal convoluted tubules, they enter the urine through a different process, though, not up here, filtration, but through active transport. All right, and then all of the urine finds its way into the collecting duct. And the collecting ducts deliver urine from the nephron tubules to the renal pelvis, which is the site that they all converge. Right? So the renal artery, again, we already did this one, brings the unfiltered blood into the kidney. And then the arterial. So now we're looking at the vascular components. I don't want to do the ones that we already did. Come on. Oh boy. Kind of glitchy now. After arterial brings unfiltered into the glomerulus. And the glomerulus is a network of capillaries between the glomerulus and the glomerular capsule. We have filtration. Let's add that. The efferent arteriole brings that filtered blood from the glomerulus to the peritubular capillaries. Peritubular capillaries deliver blood to the other components of the nephron. So that includes the proximal convoluted tubules, the loop of Henle, the distal convoluted tubules. And from there, the blood drains into the renal vein. And it's at this point that the blood is completely filtered. And it's in the renal vein that we're bringing the filtered blood back into circulation. But we're not going to write that because we already did. So let's go down to the next page. And parts, and we have this model. Normally, this model would be on your lab tables. And it looks like some of these things have been labeled already, but we can go ahead and label the rest. So here we see one nephron. This is the glomerulus and the glomerular capsule. This would be the proximal convoluted tubules that extends down to the loop of Henle, the descending limb, and then the ascending limb, which leads to the distal convoluted tubules that delivers the urine into the collecting tubules. So let's go ahead and label this. I'm going to abbreviate proximal convoluted tubules. I'm just going to say PCT. And of course, this isn't going to allow me to do that. What? Come on. Let me go back over here. Yeah, let's go PCT here. All right. And down here we have the loop of Henle. And if we follow this one, the glomerular capsule to the proximal convoluted tubules, loop of Henle, this will take us to the distal convoluted tubules. So let's fill that in, DCT. Then everything drains into this, which is called the collecting duct or collecting tubule. Right. Sounds pretty good. Moving along. Oh, no, I don't want that. All right, let's go down to the next page. All right. So this is just a different angle. You could see that unfiltered blood is in your circulation would enter into the kidney through the renal artery. So now this unfiltered blood is inside the kidney. Well, eventually it would find its way into a smaller artery called an arteriole, and specifically the afferent arteriole brings that unfiltered blood into the glomerulus. It's inside the glomerulus that the blood would be spinning around and around and around. And because of this centrifugal force of this blood that is spinning, it's causing the contents of the blood to be expelled. Once the contents of the blood is being removed, it ends up in this little cup-like structure, which is called the glomerular capsule. It's from here that this filtered blood now leaves and enters into the peritubular capillaries. These peritubular capillaries would go over the other tubules of the nephron, starting with the proximal convoluted tubules, where the good items would be put back into the blood, and then going down into the loop Henle and then the distal convoluted tubules before exiting the blood completely filtered. We have three important processes that happen. Filtration is the first one, and this is happening at the glomerulus. Well, it's between the glomerulus and the glomerular capsule. Between those two. And what is the direction, the movement of materials? While materials are moving, I'll make a new one. All right, let's just write it over here and we can move it. The materials are moving from the blood to the nephron. And what is an example of things that are moving? Well, let's see, urea, water, salt, glucose, amino acids, just to name a few, right? All right. And our next process would be reabsorption. This is where the blood is going to be taking some things back. So this happens at the proximal convoluted tubules. And what is the direction of the flow? So we could write this all in like this. Probably be easier. Well, the flow is now it's from the nephron back to blood. And what are some of the things that are going to be moving? Well, we don't want to put the urea back. We don't want to put the salt back, probably the water. If we have enough water and the salt balance is fine in the body, we'll let that go. But we do want to put all the glucose back. So we're going to say glucose is getting put back. And we're going to say that the amino acids, we want to let those go. Those aren't wastes. All right. With secretion, where is this happening? This is happening in the distal convoluted tubules. And the direction of the flow is, again, from blood to the nephron. And what is the blood adding the urine while it's dumping off some last minute items, larger wastes, drugs, let's just say. Simple. All right. So three major processes happening in different parts of the kidney. All right. And now what do we have? We have this little puzzle. We're going to trace a molecule of urea as it exits in the urine. So urea is the main nitrogen-containing waste material found dissolved in the blood, and it's excreted by the kidney as a component of urine. So pretending that we're a molecule of urine, we're in the blood is going to enter the kidney through the renal artery. And then from there, we're going to end up into the afferent arteriole, and that is going to bring us to the glomerulus, which is that circular arrangement of capillaries. So now we're going to be leaving the blood. We're going to enter into the glomerular capsule. We're going to be one of the components of the filtrate. So from the glomerular capsule, we're going to drain into what's called the proximal convoluted tubules. And then we're going to be directed down into the loop of Henle. And then from there to the, the distal convoluted tubules. Let's put that over there. And then to the collecting duct. Which will send us to the renal pelvis. The renal pelvis will direct us to the ureter. And the ureters will bring us down to the urinary bladder. And then from the urinary bladder, we go into the urethra. And then that's going to deliver us outside the body. All right. So this question is asking us, which blood vessels carry blood rich in oxygen? Well, that would be that it's in red, so we know it's oxygenated blood in there. So that's the renal artery. Right. Here's your answer. Oh, it didn't work. Whatever. And then which blood vessel carries blood less than oxygen? Well, that would be deoxygenated blood, so that's a renal vein. Which blood vessels carry blood rich in waste? Well, the renal artery has a lot of waste. That's why it's going into the kidney. And then the blood that has less waste would be the renal vein. It's because it's already filtered as it leaves the kidney. All right, so moving on. So assuming that glomerular filtration is 180 liters per day, obviously if 180 liters of waste are getting taken out of your blood per day, well, that's 92 liter bottles worth of waste. So obviously we're not expelling that much from the body on a daily basis. Most of that gets reabsorbed. Only about between 1 to 2 liters a day is expelled from the body. So we're going to look at each individual substance that is filtered into the filtrate, compare it to the substances that are actually excreted as urine, and we're going to determine whether or not each substance is either totally absorbed, mostly absorbed, partially absorbed, or not absorbed at all. And beginning with water, we could see 180 liters here have been filtered, but we only excrete 1 to 2 liters. So that means 178 liters is reabsorbed. So in this box, we're going to put an M for mostly. All right. Glucose, 162 grams, is filtered, but zero is excreted. 0 grams are excreted in the urine. So in this box, we're going to put a T for totally. All right. And so what I want you to do is, go through the rest and keep doing that. All right. You could do that on your own. If we were able to do a paper test strip type urinalysis, there's some things that are normal, normally found in the urine, and there's other things that should not be found in the urine. A couple things that we should not see are blood, proteins, and glucose. All right. If we do see blood in the urine, remember red blood cells are, even though they look small, actually too big to be leaving the blood through the walls of the glomerulus. So if there are whole red blood cells in your urine, that's a sign that the blood pressure is too high, forcing the red blood cells to leave the capillaries and enter into the filtrate. So both blood and protein, let's say no. Those should not be in the urine. And if they are in the urine, both of these would be a sign of high blood pressure. So let's add that. Glucose should also not be in the urine. If there is glucose in the urine, that is a sign of diabetes. Right? I'll keep doing that. Right, moving on. Right. Specific gravity is a measure of how concentrated solutions are. It normally ranges from 1.000, so that would be pure water. As the solute concentration increases in solution, that number goes up. 1.035 is pretty heavily concentrated, and that's the normal range that urine should be. Normally, when you wake up in the morning, your urine is more concentrated because you haven't been drinking anything all night long while you're sleeping. But however, if you're drinking water all day long and you're urinating frequently, it's going to be coming out more dilute, so closer to the 1.000 side of the specific gravity. Under what conditions will the specific gravity of urine be at the low end? So this is when you are hydrated. We are hydrated. And under what conditions will it come out high side? Obviously, when we are dehydrated. We are when we first wake up in the morning. Specific gravity reading will be higher than, right? And the color will be darker too. pH of the blood is asked to stay within a very narrow range, something like 7.35 to 7.45. However, the pH of the urine has a broader range. It could be anywhere between 4, which is acidic, to 8, which is alkaline. Here we have another picture of the blood. I'm not going to fill these in because I feel like, a picture of the kidney. I'm not going to fill these in because we kind of feel like we already did this, but this is the cortex on the outside. Deep to that is the medulla. Deep to that is the renal pelvis. And then that leads to the ureters. So you could fill that out. All right, continuing on. We have our microscopic observation. So what we're going to do is you're going to go back to the lab atlas, look at that high power view, and you're going to label the glomerulus, the glomerular capsule, and also that simple squamous epithelial cells that line the walls of the glomerular capsule. Input here are the eyepiece. I want this done using the power view. So the ocular is 10x, the objective lens 40x. And it's total magnification 400x. Right? And for the notes, what I would like is you to estimate the diameter of the glomerular capsule. All right. That's pretty good. Remember to use units. We're going to be using micrometers, right? So again, if we go back to that lab atlas, this is what we're looking for, right? This is our high power view. All right, so make sure you label the glomerular capsule, the glomerulus, and those simple squamous epithelial cells that line the walls of the glomerular capsule there. Right? All right. That's helpful. Again, this information will be on the next lab exam. So good luck with that. If you have any questions, you could email me and I'll get back to you. Okay. Bye.